Good afternoon. Silent disco talking is a new one to me, so apologies. I'm not used to the feedback. But slight change of bill. My name's Charles Weatherstone. Our founder and CEO has got a little bit of a sore throat, so is croaking. So I've been drafted in at the last minute. But it's my absolute pleasure to really introduce to you Sisaf, our technology, and really what a sensational investment opportunity Sisaf offers. Why do we say the biotech equivalent of the microchip? Well, silicon enabled the IT revolution. It's unique properties of silicon. Our invention, our platform in technology, is really the first fully bioabsorbable silicon, which has really unlocked a revolution in healthcare and a revolutionizing drug development, particularly in gene therapeutic delivery. So, who are we? We're a UK-based biopharma. We're based in Guildford, just south of London. We have a very well-protected, unique platform technology. We're partnered. I should highlight areas, but we're partnered with leaders in the field in personal care, in gene therapy. We've generated significant amounts of funding through EU grants, so we've passed some significant scrutiny. We are going to make a major impact. We've got a significant breakthrough opportunity in the delivery of gene therapy, an area that's held back currently by viral vectors and about the cost, immunogenicity, and CMC issues that are associated with them. And I'll describe some of the data that we have there. We also have a very broad pipeline of both 505(b)(2) technologies, and in personal care, some groundbreaking innovations that really address some of the environmental issues of using plastic microbeads. We've got an expert team of around 35 people, majority are PhDs. And we have our own labs, manufacturing, and bioanalytical center, really so we're a self-contained unit and not reliant on the outside world. That really allows us to accelerate our development. Finally, we're after money, like a lot of people here. We're in a Series B investment round of around $30 million and of a pre-market valuation of $180 million. This is really to drive our gene therapy portfolio and truly earn the position of the preeminent non-viral vector in that space. So a little bit of visioneering and imagination, but these are all capable with our technology. A single eye drop to cure genetic eye disease, where injections are the exception rather than the rule for dermatological conditions, and microbeads that are fully biodegradable and bioavailable, avoiding environmental issues with our use of personal care products. To go into just a very simple summary, what's the problem we're dealing with? The majority of blockbuster drugs are from biologically derived. Typically, biologically derived molecules are huge. They have significant negative charge, creating electrostatic repulsion and a lack of adhesion to cell walls, mucosal membranes. And they're also extraordinarily unstable. They can denature both outside the body and within the body. I've just used an example here. This is a CRISPR-Cas9 gene editing molecule. Typical size is anything between 150 to 190 kilodaltons. So these are not small, they're very complex, and they're very sensitive molecules. But, really, drug development technologies haven't moved on. The majority of drugs in pipeline are delivered by injection. This is a patent from 1845 for one of the first hypodermic needles. So we're using technology that's getting close to 200 years old in the majority of our new chemical entities. Viral vectors. The majority of drugs that have been approved, gene therapeutics that have been approved, and the vast majority of pipelines are reliant on viral vectors. Of course, they have significant immunogenicity issues. They have manufacturing challenges, and they're very expensive and quite limiting to small companies with gene therapeutics to have access to. Liposomes and polymers, incredibly useful but limited entrapment, tendency to leak, to break, and can't control the release of drug. And lastly, inorganic nanoparticles. They don't biodegrade, they accumulate. So there's FDA pathways specifically for inorganic nanoparticles that cannot degrade. So what do we bring to the world? As I said, we're focused on silicon. Elemental silicon, it's the second most predominant element on the planet behind oxygen. So we'll never run out of it. It's a plentiful supply, and it's a pretty cost-effective. It's also an essential micromineral for human health. About 1.4 grams of the average human is made up of silicon. And that silicon is responsible for collagen production. It's a collagen precursor, elastin precursor, and bone mineralization. So it's biologically friendly. Very much like the microchip world, we precision nanofabricate these silicon particles. We can be very precise. This is a process of physics rather than chemistry or biology. So we produce these nanopores or etchings, and it gives us two functional surfaces. We can fill these pores with these large, complex molecules and condense them, but we stabilize them by we don't have any bonding to those, so it's non-covalent bonding, so these molecules are highly stabilized. These pores are also disconnected, so we can put two separate entities into the same system and release them at the same time. We then decorate the surface, and so we decorate the surface with amino acids. That's the bottom part of our patent that ensures 100% bioabsorption to orthosilicic acid, so it can be used by the body. The lipids close the matrix, close everything in, and we can adjust charge. Receptor-mediated ligands can be added. So this is a process of decoration. We've got significant evidence around crossing stratum corneum, cornea, really quite formidable biological barriers. And most importantly, as the particle hydrolyzes, it releases its product, it releases its payload. But we can control that rate of hydrolysis from hours through to days, even weeks, giving these drug compounds time to operate, but also reducing toxicity to cells. So, in essence, we've got a highly versatile, enabling technology that's made from FDA GRAS products, FDA GRAS materials, and we've scaled it cost effectively. We're commercialized in personal care markets. We're moving that way in RX and in gene therapy. So we've scaled the manufacturing, and we can really address some of those cost and CMC issues that the gene therapy market faces. In terms of competitive set, we believe we're unrivaled in terms of the versatility and the safety due to that bioabsorbability, nothing left behind. And how do we apply this unique asset? This is quite a complicated slide, but really our markets, we can access multiple billion-dollar markets. We've focused in the pharmaceutical and personal care area because of size and because of our background knowledge and understanding of these markets. And we really split these out in, we've got a very compelling personal care. We work with leaders in that field. In 505(b)(2), an accelerated pathway where we use existing APIs to reduce clinical risk, but we change their route of administration. We adjust their toxicity. But ultimately, our focus and our future is in this breakthrough area of gene therapeutics delivery. Our pipeline, it's a very full and compelling pipeline, combining both partnered and our own pipeline, so we mitigate risk to investors. We've got some fascinating areas, and I'll go into a little bit of detail just around our gene therapeutic entity. Through a knowledge transfer grant and working with the University of Ulster, we recognized that we could deliver siRNA and CRISPR-Cas9 gene editing technology to all 5 layers of the cornea. These are densely packed cells, no blood flow, a very high transit time, so it's a really challenging area to get anything meaningful into this area. We found all 5 areas had penetration and permeation. So that's fine, but what does that mean? Well, we've got very high entrapment efficiency, we showed very high transfection efficiency, and a significant reduction in gene expression. And this was using a TGF-beta knockdown, so it's going to be a regular treatment. Regular injection would be challenging, would be painful, expensive, and harmful to the patient. We can achieve this with a single eye drop every 1 to 2 weeks, and then moving into CRISPR-Cas9, that could be a cure with one single eye drop. That is, of course, repeatable and applicable to all siRNAs. So I'm just going to whip on. We've got a really strong team. They've been there, they've done it. We've got medical pioneers, commercial innovators, and some of the best advisors and leaders in the gene therapy field. I'm being waved at by the back, so we'll quickly move on. In terms of prior investment and where we're going, we've raised a total of $14.5 million to date, and we've been awarded over $3 million of EU and UK grants. Our mission at the moment to take us, this is really a pre-IPO round. We're looking for $30 million on 180 million valuation to truly drive that gene therapeutic entity into human, to commercialize our 505(b)(2) portfolio, and really to exit in primary care and generate some high-value exits. So an enormous amount of milestones to achieve, but within the funding window, our mission is really to gain access to US capital markets and mature Sisaf, so we're prepared and ready with clinical success, commercial success, a mature business, and a massive amount of news cadence to keep public investors interested. So a final word is really, again, silicon drove the digital revolution. I think with this innovation, we have an opportunity to drive a healthcare, particularly a drug development revolution with Sisaf. Thank you. Any questions?